Good morning, everybody. So, this is Anuprita Chopde, student of FYM Farm, Department of Pharmacology. Would like to present a research article as a part of Journal Club activity, guided by Dr. Praveen Badhe sir and co-guided by Rutuja Mule ma'am. So the title of my research article is Gene Identification and Potential Drug Therapy for Drug Resistant Melanoma with Bioinformatics and Deep Learning Technology, which was the co-author of this research article is Mr. Yingbing Zhu, and this article was published in 23rd July 2022. So, what basically is text mining? So, the text mining makes it possible to collect the disease gene association automatically from large volumes of biological literature. Further, identification of genes associated with drug resistant melanomas can be done using the text mining tool PubMed to Ensemble. Gene screening was done using Go and KEGG pathway enrichment analysis. The PPI, that is the protein protein interaction network, was constructed using the string database and cytoscape and also the Davis database. Further, the very important tool, if we look at this flow chart, JPA. Now, what is JPA? JPA was used to perform survival analysis. The full form of JPA is uh, gene expression profiling interaction analysis. It allows the users to apply custom statistical methods and thresholds on given dataset to dynamically obtain differentially expressed genes and their chromosomal distribution. Now, JPA fills in the gap between cancer genomics, big data, and the delivery of integrated information to the end users, thus helping to unleash the value of the current data sources. So how we can do the survival analysis? For this purpose, we can also use Kalpan-Meyer curve. Now, what is Kalpan-Meyer curve? So it is defined as the probability of surviving in a given length of time while considering time in a very small intervals. Now there are three assumptions mainly used for Kalpan for this kind of analysis. Firstly, we can assume that any time patient who are censored have the same survival prospects as those who continue to be followed. Secondly, we assume that the survival probabilities are the same for subject recruited early and late in the study. And thirdly, we can assume that the event happens at the time specified. This creates problem in some conditions when the event would be detected at regular examinations. Now, in short, Kalpan-Meyer estimate can also be called as the product limit estimate. So, for further, if we can see, the drug target interaction has also been done, and for this purpose we have used the pharma project. Now, what is pharma project? Pharma project is a database which can be used to inquire about drug target at hub genes. Now, each hub genes generated drugs list target on it. Lastly, deep purpose is also achieved by and for this, from this, drug target identification prediction can be done. So, in the conclusion part, we can say that the relevant genes of drug resistant melanoma, based on the technology of mining, 433 genes were found by using the PubMed to ensemble. Then, further, most statistically significant process in the GO-KEGG analysis was done. And further, the PPI interaction, that is the protein protein interaction network, was built by using string, cytoscape, and other tools such as Davis, and where 27 genes were screened out by the M code. Further, the JPA was used to establish six genes. Statistical difference in survival analysis was done by using the JPA, and further 16 targeted drugs were identified by using the pharma project. Now, what is deep purpose? So deep purpose was used to identify the drug target interaction prediction, and lastly 11 drugs were screened out. So this was about the article. These are the disease markers, the hub genes which were selected by cytoscape and encode. And yes, these are the results under this article. Thank you.